Hi everyone, this is Dr. Chandana Lakshmi, working as professor in the Department of Chemical Engineering, AE College of Engineering Autonomous, Andhra University, Visakhapatnam. Today I am going to tell you about the microbial degradation of phenol using bacteria Pseudomonas aeruginosa. The Environmental Protection Agency, that is EPA, has listed 23 organic pollutants. Among this 23 organic pollutants, phenol is one of the rarer organic pollutant which comes from various industries like steel industry, pharmaceutical industries, dyeing industry, textile industry, varnish industries, etc. There is a necessity to meet the standards of the effluents which is given by the EPA, as it causes hazards to both the flora and fauna and also the plants and also to the animals. From where do we get the phenol? This is nothing but by the natural sources, that is by the decomposition of the organic materials and the forest fires. And as I said before, like industries like petroleum refinery, pharmaceuticals, coal refinery, paper and pulp, varnish industries, steel industries, etc. There are why is it necessary to remove these phenol? Yes, there is a necessity. Why? Because it causes both the acute and also the chronic effects. Acute effects like coma, shock, collapse, and sometimes it may also lead to death. And chronic effects like swallowing, diarrhea, and also vomiting and skin diseases, etc. So there is a necessity for the removal or at least for the treatment of the phenol, like the methods like physiochemical methods and biological method. Coming to the physiochemical methods, we have like chlorination, advanced oxidation processes, adsorption, solvent extraction, coagulation, flocculation, reverse osmosis, ozonization, photocatalysis, electrolytic oxidation. By using these physiochemical methods, a lot of research work has been done. But there are certain limitations for these physiochemical methods, like incomplete phenol removal, and also it requires very high reagent and energy requirements, that is high temperature and also high pressure, which leads to a very cost which leads to high cost. And also in addition to this, there is also a secondary product also is produced, which also has to be again removed from the environment. So in order to avoid these limitations, we have an alternative like called the biological tool, which is nothing but biological method, that is by using bioremediation. We have lots of biological methods. In this study, I have gone with the bioremediation method. Bioremediation is an alternate, as I said, it is an alternative to the physiochemical methods. So it has an advantage like it is a low cost process, and it is also a recent process, and it is also an eco-friendly process. It uses the microorganisms. Bio, it is nothing but life. So it uses microorganisms for the removal of this phenol or the any hazardous pollutant. So in this biological method, the main parameters to be considered are the temperature, pH, the moisture content, nutrient availability, contaminant toxicity, contaminant concentration, and oxygen availability. And with respect to both the phenol and with respect to both organism, also there should be a study. Finally, I would like to conclude, or I would like to close my presentation saying that the parameters or the parameters like the growth of the microorganism and the conditions in which it grows, like temperature, pH, and the concentration of the substrate, all has to be considered. And at the same time, we have to measure the phenol concentration at which level, like at which optimum conditions for the growth of the microorganism, at the optimum conditions, what is the phenol concentration, what is the removal percentage of the phenol concentration. So at the optimum conditions, the removal of the phenol concentration will generally be high. Thank you.